Again, welcome to Statistics. This is our first lectures in our course textbook, chapter 2, and it's unit 2, lecture number 1. So the first thing we're going to cover is the scientific method. So the procedure for scientific method is, first thing we need to do is gather information about the phenomena that we want to study. So we need to get some data or gather information about the phenomena that we want to study. Now, based on the information that we gathered, we can formulate the preliminary generalization or hypothesis. We can also formulate the study question or the research question. Then from there, we can collect further data to test the hypothesis. Normally in statistics, we are going to use the sample of the data to test our hypothesis. Hypothesis, we have the null hypothesis, which is the claim. Then alternative hypothesis will go against the null hypothesis. So example, if we want to do a studies or we want to do a research about weight problems in US, and we know that in 1970, the average weight of adults in US is 200 pounds. Now, 50 years later along the way, we want to test if this 200 pounds remains the same. So our null hypothesis will be the 200 pounds that we know in 1970 is the average weight of adults in US. So now our alternative hypothesis, either we go against it by saying that it's less than 200 or greater than 200 or not equal to 200. Not equal to 200 is called a two-way testing. We want to test both less or greater. So after that, the solution that we get, or during the testing, the answer that we get or the result that we get will become a law. So here we say that the final step 4, if the data and other subsequent experiments support the hypothesis, then it becomes a law. So again, these are the 4 general steps in scientific process or method. First, we need to gather information about the phenomena that we want to study. Now, on the basis of the data, we are going to formulate a preliminary generalization or hypothesis. Then we collect a further data to test our hypothesis. Now, if the data and other subsequent experiments support the hypothesis, then it becomes a law. Now, during studies, especially experiments, we have something we call the confounding variables. Confounding variable will be the extra variable that are not accounted for during experimentation and can cause the result to be skewed. So the key word is that confounding variables are not accounted for. It can be the third or extra variables. So a very simple example would be if we are doing studies and we want to find out the correlation between ice cream sales and shark attack, there can be an association. But what will cause the association between ice cream sales and shark attack rate will be the weather. So we know in Florida during the summer, there's a lot of shark attacks because the ocean gets warm and the sharks come close to where human beings are swimming or whereas during that summer, ice cream sales also goes up. So here we have a confounding variable, a third variable that causes the relationship between shark attack and also ice cream sales. Also, a confounding variable is something that we cannot remove from our experiment, but we always do our best to minimize it. So we have something called the residual confounding variable. Residual confounding variable will be the confounding variables that remains in our experiment after we try to eliminate or minimize all the possible confounding variables, identify them and remove them. Confounding variables are not good in research process because they can change the result of the outcome of the experiment. We also have what we call the modification effect. These are also variables, but these variables can either increase their outcome or decrease the outcome. So it's very good. For example, we are doing an experiment to find out if there's a relationship between cigarette smoking and lung cancer. Well, we know that if you smoke cigarettes, the chance of getting lung cancer is high. But there are some people that they only smoke cigarettes when they drink coffee. So this means a coffee can be a modification effect. And the more coffee I drink, the more cigarettes I smoke. The chance of getting cancer is high. Or maybe there's a special drugs that when I smoke a cigarette and I take that drugs, it will clean my lungs, which will make it possible not to get a lung cancer. That also can be the modification effect, a variable that can reduce the outcome or increase the outcome. So next is the decision-making method. The procedure is first, we need to clearly define the problem. Anytime we are doing a research, the main goal of a research is to solve some problem or to find a solution to a problem. So we always need to clearly define the problem and any influential variables. Then we decide upon objectives and decision criteria for choosing a solution. Then we create our alternative solutions. We compare the alternatives using a criteria established in the second step, which is deciding upon the objectives and also decision criteria for choosing a solution. So this will be a process of making a decision. Now, next we implement the chosen alternative. Then we check the result to make sure the desired results are achieved. Now, collecting the data is very essential in any research because, again, most research are now based on data-driven. So essentially, there are two ways to obtain data. Either we can collect the data by observation or by control experiments. We can collect data by doing experiments in a laboratory based on whatever input we give, if we are using biological organism or whatever components we are using for the experiment. Again, we can generate data based on the behavior of whatever experiment we are doing. So data collection method is related to the nature. So again, data collection method is related to the nature of the problem to be solved and also the ethical and practical constraints of collecting data in some environments. And there are so many tools in collecting data. So the conclusions suggested by statistics can be no stronger than the quality of measurement which produce the stat evidence. Also, fuzzy or confounded measurement must be produced fragile conclusion. Again, we will go detail on this when we start inferential statistics. So when we are doing experiment also, we have two important variables. We have what we call the response variables. Response variable normally measures the outcome of an interest in a study. So response variable will be, we can say the term dependent variable. So if I'm doing an experiment between S and Y. If S will determine Y, then Y is a response variable or dependent variable. So our input is X. For example, cigarette smoking and lung cancer experiment, the input will be cigarette smoking because cigarette smoking will cause the lung cancer. The more I smoke, the chance is high. The less I smoke, the chance is low. So in any experiment, we always have a response variable which will measure the outcome of the interest in the study. In this case, it's a lung cancer. Then we also have an explanatory variable. The explanatory variable will cause or explain changes in response variable. So the more cigarette I smoke, the outcome of lung cancer can be high or low, depends on the number of cigarettes or how long I smoke. So an explanatory variable will cause or explain changes in the response variable. Now let's see example 2.1.1. Here they say suppose, so the example here said, suppose a new species of tomato has been genetically engineered to increase yields. How can we measure whether the new species produces higher yields? So the solution here is said, in this experiment design, the plots of tomatoes will be divided into two groups. One that contains the new species and other that contains the unaltered variety. So the plots containing the genetically engineered variety will be a treatment group, and the plots containing the standard variety will be the control group. And normally this type of experiment, we also call it case control experiment. So for example, if I have any studies I want to do, or I want to test an efficacy of new drugs, I may try to get two groups of people. I may give the drug to one group, and maybe I may give a placebo to the other group, and I'll see the outcome of both of them. So this is a general experiment. We have a tomatoes, we genetically engineer it to yield more. But now, how do we know if the procedure we follow give us the result, the desired result? The good way to know this is we can divide the tomatoes into two: the ones that will grow naturally and the ones that are genetically engineered to increase yields. Then we see what will be the result. So if the experiment is properly performed, any change in the response variable, which is the yield, can be attributed to the experimental variable, which will be the again genetic engineering. So genetic engineering is the independent variable, the input, and the output to be response variable, which is more yield. The yield will either increase or decrease. We want to know. So variables like rainfall, amount of sunshine, or type of soil can affect the yield of tomatoes. So these are some of the variables. Maybe we may call some of them a confounding variable. It depends, or modification effect. This variable must be controlled in the experiment so that differences in yield between the plots can be attributed just to genetically engineered species. This is very important almost in every studies. So in these studies, it's about tomatoes. We want to know if the tomatoes genetically engineered will yield more than naturally. But we also know that rainfall, amount of sunshine, and even the type of soil can have effect on the tomatoes yields. So we need to control these variables. So one possible method of controlling for these variables is to create a small plots in the same field, all with similar soil types. So we can see that we have plot 1 up to plot 16, the same field, but we create different. Then several plots of each type of tomatoes are planted. Now each plot contains only one type of tomato. Rainfall is measured and supplemented with irrigation to assure that each plot has the same amount of water. By controlling for these variables in this manner, again, we can say that any differences noted in the yield, that is the response variable, between the genetically engineered variety, the treatment group, and also unaltered variety, which will be the control group, is due to genetic improvement, which is our input explanatory variable. Another example here, we say, does an SAT preparation course improve performance on the SAT? Again, we're going to perform experiment. We need a data. We need an input. So here, a group of high school students take the SAT score, Then they are given an SAT preparation course. They retake the SAT again. If the group's second SAT score performance improve, then it may be related to the SAT preparation course. The explanatory variable, which is our independent variable, will be the preparation of the SAT, the course. And of course, our response variable will be the score, SAT scores. So before, the control group, high school students that have taken the SAT exactly one time, the value of the response variable is the student scores on their first SAT. Now the treatment is the high school students are given SAT preparation course. After that, they are going to take so now the same students are the treatment group and they take the SAT score again. Now we compare the two results, compare the students' performance on the SAT before and also after. Then there we can make our conclusion whether our explanatory variable courses, which is the SAT preparation course, improve the SAT score. So in this experiment, there's only one group of students. Suppose that there were two groups of students, one that has taken the course treatment group and other that had not taken the. Could you assign any differences in the results solely to SAT preparation course? Of course, no, because there may be other variables. Maybe the intelligence of some students, there's two differences. And also family issues, backgrounds, a lot of variables will be considered. But this is a perfect way. We are using the same student. Actually, in statistics, we call this the pair test, pair sample. A pair sample means in this case, I have a sample of students, I let them do something, then I give them some treatment, and I let them do something again. So the same sample, the same size. Independent sample means I can collect two different samples from even two different populations. The size doesn't matter. So because of potential differences in the cognitive abilities of two groups of students, this experiment will be much more vulnerable to justify criticism. And in this case, we are saying that if we use the same student, they take the SAT score, we give them SAT preparation course, and they take the SAT test again. So they took it first time, we get their score, we give them preparation, then they take. This is better than saying that we have two groups of students. One shouldn't take any student SAT preparation course, they just take the test straight. One group of students take the preparation course before they take it. We cannot justify that only the preparation course make these two tests different, because as we can see here, intelligence can be different, cognitive ability, there are so many different factors. So next we go to what is a placebos, which we use it a lot, especially when we are doing a case control experiment. A placebo will be a fake treatment that has the potential to cause a response. And normally when you are giving a placebo to a subject, you don't let them know. You let them think it's the real medicine or the real treatment, so that there won't be no bias. We also have what we call the double-blind studies. So a double-blind studies normally are used to counteract the placebo effect. In a double-blind study, the subjects are not told whether they are members of the treatment group or control group. And also the evaluators, or if they are medical doctors, they also don't know, they are not told. So a double-blind study means, again, we are doing studies, we have a control group and the actual group. We don't let the control group knows that, again, they are being given a placebo. And also the evaluators of the experiment, also we will not let them know. But a single branded means the evaluators know, but the subjects in the experiment, they don't know. So bias also is very important. So bias will be the tendency to overstatement or understatement the value of certain population parameter. And bias means, again, an error in our experiment. So this will be the conclusion of our lectures here. Again, the main goal of these lectures is to get the basic concept of a research process experiment, because again, in statistics, we always do experiments. So first in statistics, we collect the data. If possible, we prepare the data, clean up the data from errors, store the data. Later on, we analyze the data using any statistics techniques. Then from there, we get our results. We apply the result to the population. Now, the steps that we follow to analyze the data, example can be a hypothesis testing or estimation or regression analysis. We will come detail on that in our future lectures. So again, wish everybody the best and thank you.